மைக் மட்டும் ஆன் பண்ணிக்கிறேன். அவன்கிட்ட சொல்றேன் கொஞ்சம் ஆளுங்க வரட்டும் அப்புறம். இல்லை. Hi. So we'll start the session once a few other students join. Please stay on the call. Hi Surinder, hi Ignatius. We'll just start in a few minutes. I hope you can see my PowerPoint that's being shared.
Yeah, yeah.
Okay,
PowerPoint is visible.
right, thanks. Okay, let's start. Yeah, this video will cover some assignment problems, sample problems related to the lectures for week 6. So the first question is, in the isotopomer distribution vector, a 3-carbon molecule is represented as 001 bin. This implies that the option A, the molecule is labeled with a single 13-carbon isotope in the first position. Option B, the molecule is completely labeled. Option C, the molecule is labeled with a single 13 carbon isotope in the last position. And option D, the molecule is completely unlabeled. You can take a couple of minutes time and then maybe with we can start with discussing what an isotopomer distribution vector is and how molecules are represented, which will help us find the right answer. Yeah, does anyone want to share what they understand about isotopomer distribution vector? Uh, Ignatius, Raymond, Peter, or Surendra? Anyone?
Yes, sir.
Yeah,
Am I audible, sir? Hello.
yeah, you're audible.
Yes, sir. So, according to my understanding, isotopic distribution vector basically represents distribution of the labeling in a particular molecule.
Huh.
So, since the question implies that there are three carbon molecules, and 001 implies that each of these molecules could either be labeled or unlabeled.
Hmm.
And since the third digit here like denotes one, so I'm going to go with option C. So the
Right,
molecule is labeled at the last position. I'm going to go with that. I'm not entirely sure though.
right, right. Surendra?
Uh, I don't have idea about that.
Okay, okay, right. I'll just explain that. Yeah, so adding on to what Ignatius said, yeah, so this vector called the isotopomer distribution vector, it's usually generated with the intensities of the different possible isotopomers that one can get when they are doing a labeling experiment. So an isotopomer is basically it gives you what are all the possible positions that can be labeled for a given molecule. Let's say there is a two carbon molecule. There are different possibilities, like both the carbons could be labeled, or the first carbon could alone be labeled, or the second carbon alone could be labeled, or both the carbons can be unlabeled. This is the set of possible molecules that can come when you have a labeled substrate that gets converted to this two carbon molecule. So is that first clear about the labeling and formation of isotopomers, Surendra?
Yeah, clear.
Yeah, that is clear. So when we get such a distribution of molecules for this metabolic flux analysis, further, it is not necessary that all these four different or all these n number of different um, labeled molecules should be equally distributed. They could be of varying intensities. So, from gas chromatography and mass spec experimental results, one will be able to obtain what fraction is fully labeled, what fraction is not at all labeled, and what fraction is partially labeled. So you will basically get intensities, and that is represented as a vector. That is the isotopomer distribution vector. So for a three carbon molecule, one can make such an isotopomer distribution vector with uh, eight different variations, starting with 000, then 100, 010, and 001. What is being asked here is the 001 bin, which means that in the last position, the molecule is labeled with a single 13 carbon isotope, which is what Ignatius had mentioned. So that is the right answer.
So what is the meaning of this bin?
Bin is nothing but how do you, uh, okay, let us take in a particular distribution, you will have ranges like 0 to 25, 25 to 50, 50 to 75, 75 to 100 if the range is between 0 to 100 and if you divide it into 4. So now each of the particular set of values corresponding to a particular range, like for example 0 to 25 or 25 to 50, each of these are called bins. So you can call this as divisions in a particular graph. In with respect to this isotopomer distribution vector also, you will have a particular intensity associated with this molecule which has the 13 carbon isotope in the last position. So basically that acts as a bin. It's a mathematical term that people generally use when they try to segregate data into particular ranges. Is that clear?
Yeah, yeah.
Okay, okay, right. Yeah. Now the next question. What is the limitations of 13 carbon steady state metabolic flux analysis? Option A. Achieving isotopic steady state is not always possible. Option B. It's useful to study cellular metabolism. Option C. Quantify the relative abundance of isotope labeled atoms. And option D. All of the above. So let's not rush through the answer. We'll take a few minutes and first go option by option and see if each of the option is suitable or not. We'll take a couple of minutes and then answer this question.
So, can we discuss this?
One minute. So we can start with option A. You can first say if you think how steady state can be achieved and whether that is the reason why it is not possible or if it is possible for 13 CMFA. We can start with that and then we can see if Ignatius can answer for any of the other options. We can take turns and try to say why a particular option is right or not. Okay? Yeah, Surendra. Or if
So,
you want, you can talk about any other option as well. It's up to you. We'll be discussing all the three.
so C13 labeling can be used to study the cellular metabolism, and it
Hmm.
has been proved in the previous studies also, like
Hmm.
Kelvin cycle.
Hmm.
They have been studied by the labeling of the carbon atoms.
Hmm.
So this is not the limitation of the. I think this is not the limitation of the MFA C13
Hmm.
MFA.
Hmm.
Also, quantification of the C13 molecule uh, we can do with the help of these techniques with accuracy. Like GCMS is there
Hmm.
and
Hmm.
NMR is there. They can find the relative abundance accurately. So,
Right.
but achieving the steady state, it is not always possible
Ha.
of isotope. So, I think first is the limitation.
Right. Right. Okay. And you, Ignatius, do you want to comment on this?
Not exactly, but yeah, I'm having confusion with the option A. I would agree with the other two points. So yeah, it can be used to study cellular metabolism as the labeled carbons usually go through a pathway and you can keep the track of them. And
Hmm.
the relative abundance can be measured through techniques like GCMS and NMR.
Right.
I'm not sure about option A though.
Okay. Yeah. So, right, like both of you had mentioned, one thing that I would also want to comment on is that although we say that the TG steady state MFA can study cellular metabolism, it has a limitation on the number of pathways that can or how branched a pathway it can study. So, what people usually take up from 13C steady state MFA is they try to limit the number of metabolites and the reactions, and they basically one can't study the entire pathway that's within a cell or in all the reactions, 3000 reactions within the cell. So that has to be kept in mind. Although people mention that it can be used, people mention that it can be useful to study cellular metabolism. It has to be understood that not all the reactions at the same time can be studied. Only parts of the reaction can be studied. That too, based on the divergence to amino acids. And yes, option C, which is quantifying the relative abundance of isotope labeled atoms, that uh, only because we are able to quantify the relative abundance, the study its 13C MFA itself is possible. So that can't be a limitation. But whereas achieving isotopic steady state is not always possible. So this is something that has to be taken into consideration because, firstly, in cultures, cell cultures, when you grow it in large volume, ensuring that steady state is happening itself is dependent on other factors like the flow rate in which the media enters, the flow rate in which the cell consumed or spent media exits. There are different culture settings like batch culture, continuously stirring reactors with different processes. So there, achieving steady state itself is difficult, although it is doable. So, but achieving isotopic steady state is close to uh, dif- more difficult since the amount of substrate that one would have, like for example, labeled glucose would be very limited. So with the limited amount of labeled substrate, it's very difficult to achieve steady state. So that is highlighted as the reason, and that's the answer. Yeah. So
Steady state means every possible pathway.
no, no. Okay. Here, steady state means what there is no accumulation of any of the metabolites in the cell and in the culture vessel. So you have a culture vessel where you have your media and your cells, correct?
Mhm.
So you will be supplying some amount of this labeled substrate, right? You will be continuously supplying that in many cases of cultures. When you continuously supply them, you should ensure that it enters the cell, then gets converted, and then exits the cell, and then goes through the media, and then the media also gets out, has an outlet. So basically, there is no accumulation of this labeled metabolite. That is what is meant by steady state.
Mhm. Okay.
Was that clear?
Yeah.
Okay, right.
So,
Yeah,
I want to ask if, for example, we supply 13 glucose
huh,
to the cell
right,
and cell is performing the glycolysis.
right,
Okay.
right.
So, and pentose phosphate pathway is not occurring at that time.
Okay,
So, C13 carbon, it will be distributed in the glycolysis and TCA, na? Not in the pentose phosphate pathway.
okay,
So,
okay.
in this way also, steady state is not achieved. Na? Means C13 glucose it is not uniformly distributed in every possible pathway of the cell.
No, no. So we are not talking about whether C13 that is entering the cell is being distributed through all the pathways. What we are saying is C13 that is entering the cell, it is whatever C13 is there, it is being distributed. There should be no C13 that is in entering the cell, but that is not being used by the cell.
Okay.
That is what we are saying here by steady state. Because if that is the case, you will continuously supply C13. The cell will take it, but if the cell is not using it, you will think that the cell is using it and it is producing different isotopomers and you will have all those matrices. But in reality, only a fraction of that will be being used by the cell. When a fraction of it is being used, but you account for the entire amount of isotope uh, being given to the cell, then it would not equate. That is where you will have inequalities. So the entire calculation that goes behind would not give you the right flux values. That is why we always aim to achieve steady state distribution.
So
Yeah,
yeah, so according to what you're saying, in the steady state,
huh.
whatever is being taken in will be utilized completely. That's what steady state is, right?
It should be, so the thing is it should either be converted to other smaller products or larger products. It should be incorporated. Or if it's not being incorporated, it should exit the cell. Whatever is entering should exit the cell. There should be zero accumulation within the cell.
Okay, sir.
Yeah.
And so this is not possible due to constraints, correct?
I could not understand what you mean by constraints. Can you repeat that? Can you like elaborate on that?
Like depending on the environment that is the cells are considered in,
Yeah.
so maybe some environmental factor that isn't, making this. metabolite to go through a certain pathway and it causes the accumulation or something like that. So can that be seen as a consideration for not achieving this isotopic steady state?
Yeah, that is one case, and another case could be the state of the cell itself. Like, for example, when you have too much of substrate, very limited of cell, very little cells, smaller in quantity or number, and abundance of substrate, what will happen is the cells will replicate. But after some point, once they go beyond the stationary phase, they won't be replicating. So they, even if you supply it, it might take it in. So there is a fine balance between the growth rate and how and achieving steady state. So it depends on what growth rate or growth stage of the cell is currently in. That is also another consideration that one has to uh, take into consider take into account when they're talking about steady state in microbial cell cultures. Apart from the environmental conditions in which the cell is.
Yes, sir. Thank you.
Yeah. Okay. Yeah. Huh. So the next question. Uh, yeah. In the above represented reaction map, R1 to R8 represent 8 reactions, whereas A, B, C, D, and E represent 4 metabolites. Okay, it's actually 5 metabolites. Huh. Using the logic of pull and push mechanism of metabolic engineering, identify the reactions that can be eliminated to maximize the production of metabolite C. So here they have given you a reaction map where R1 is entering the cell and it gives A. Now A is getting involved in 2 reactions. Reaction 2 R2 where it gives B, and reaction 4 R4 where it gives D. Now metabolite B gets converted to metabolite C through reaction R3, whereas it gets converted to reaction E in the presence of metabolite D through reaction R5. Then E exits the cell with R7 and C exits the cell with R8. So now we have to identify which all reactions we can eliminate here so that maximum of C can be produced. And they also mentioned this logic of pull and push mechanism, which was discussed in the last lecture of this particular week. So we can take a couple of minutes and first start with what the pull and push mechanism of metabolic engineering is, after which we can try to see manual variations as to how a particular reaction, eliminating a particular reaction, would improve or decrease the production of metabolite C. We'll take a couple of minutes and then continue with this.
Should we start?
Yeah, yeah. So you can first start with what is the logic of push and pull mechanism that was discussed, and then we can take it from there.
Pull and push means push the means we have to force the precursor molecules,
Huh.
okay, to the our target molecule.
Huh. Yeah. So just to rephrase, like what you said, so we would push the upstream pathway or manipulate so that the precursor metabolite synthesis occurs. Then we'll pull it for production of
Mhm.
our downstream product of our interest. So that was the pull and push logic that was discussed. Now we can, instead of going
So
directly to the answer, Surrender, we can first discuss how eliminating a particular reaction would either improve or decrease the production of metabolite C. Yeah, you can start with any option that you want to discuss.
so in this we have to block the competitive path also.
Okay,
Okay, so here A to C and A to D. So R4 is the competitive pathway.
right.
So we have to block this.
We have to
Similarly,
remove
we have to
R4,
block the
R5.
R5 also.
Right,
Uh, So if we block the R4, R5, R6, R7, automatically it will be blocked.
right.
So C is the answer.
Okay, right. Do you have anything beyond that?
So in that for push we have to increase the expression of the flux limiting enzyme also. For example, any enzyme is not properly targeting the particular molecule to the product. So we
Hmm.
have to enhance its expression also. Flux limiting enzyme.
Right. Now,
And
sorry.
pull means we have to enhance the expression of last enzyme of the extreme path.
R3 is it?
R3,
Right,
R8, R8, R3.
right. No, okay. I was about to ask that. So, let's say we remove reaction R8, what would happen? Or if we enhance R8, what would happen? Do you have any
If
possible
we
idea?
uh, molecule will be exported out of the cell and it is desired end product accumulate inside the cell, we prefer our metabolite should be excreted out by the cell so that purification can be easily and cost effective also. And we
Ah,
don't have to kill the cells also.
yeah, like, so let's talk about the aspect in which reactions and metabolites are affected by metabolites itself, because that's what we are doing here, like elimination of reactions. So if we block reaction R8, what would happen and how would it affect usually in metabolic processes? If you have an idea, you can share about that. Even Ignatius can answer if Surendra,
So
one after Surendra. Yeah.
downstream processing will be difficult.
No,
If
no,
you block the
we can limit the down. We can consider that the downstream.
RA,
We can consider that the downstream process is not affected. Now, if R8 is not there in the system, would it affect the system in any way?
yeah, well, feedback inhibition.
Yeah, okay, that's actually what I wanted to ask. Yeah. So what I would want here is that if R8 is increased, more of the C would exit the cell, which itself would in a way drive production of C. Whereas if R8 is removed from the system, C would accumulate, and like you had mentioned, product inhibition, which usually is observed in many enzymes in many pathways, would occur where the C would limit one of the enzymes that produces or is the reason for production of C, and it would reduce the production of C. So the answer here, rephrasing Surender, is that option C, where if you reduce R4, it will prevent formation of D. If you reduce R5 or remove R5, it will prevent formation of E. So R6 and R7 will no longer exist within the cell itself. So removal of R4, R5, R6 and R7 would enhance the production of C. Yeah. Uh,
So
yeah, sir.
if there is a fifth option,
Yeah.
R4 and R5.
R4 and R5 alone. Is it?
Fifth option is R4 and R5 alone. So which will
Okay.
be the most preferred answer here?
So from what we see in the reaction network here, if R4 and R5 are removed, R6 and R7 will definitely not exist, right?
Hmm.
So only if R4 exists, A will be converted to D, and only if D exists, R6 will happen. So if R4 is not there, R6 will not happen, even if R6 is there in the reaction. You can imagine this like a knockout of a particular enzyme in step one, where there are multiple steps in the pathway. Where if the first enzyme itself is knocked out, the, the metabolite will not proceed through that pathway at all. So just having R4 and R5 is equivalent and sufficient for production of C. It is an equivalent option compared to R4, R5, R6, R7. So both are equally valid.
Okay.
Yeah, yeah, yeah. Ignatius, any doubt? Was this clear?
No, sir.
Yeah,
Clear.
okay. Yeah, yeah, that's what they mentioned here in the solution as well. That the production rate of metabolite C is dependent upon the availability of metabolite A and B. Reaction R1, R2, R3, and R8 is responsible for the production of C, whereas reactions R4 and R5 are consuming A and B to produce D and E respectively. Furthermore, the flux of R4 and R5 is followed by the reactions R6 and R7. Therefore, if we eliminate R4, R5, R6, R7, then we can get a higher flux through the reaction R2 and R3 and R8 that can increase the production of metabolite C. Okay. Ha. The next question. Consider a carbon exchange reactions R1 where A, which is a 3 carbon substrate, gets converted into B, which is a 2 carbon substrate, plus C, which is a 1 carbon product. Sorry, B and C are products. Now choose the correct isotopomer mapping matrix for A to C. Okay. And they have given 4 different options. We can take a couple of minutes and then we can start working on this problem. Yeah, so
Actually,
let
I'm not getting the answer. Ignatius can proceed. I'm
yeah,
not getting.
yeah. If we can try, yeah, Ignatius, you can try answering. If not, we'll try solving.
Oh no, sir. I don't think even I'm getting the right answer, so we could go away with it.
Okay, okay. First, I'll define what an isotopomer mapping matrix is. Then we'll see how we can do it for this particular case. Okay, okay. So, just like any other matrix that we would draw, this is a matrix that is used to understand how a particular isotopomer, meaning a substrate with different labeling fractions, can give rise to a different product. Okay, so what I mean by here is we have a substrate A which has three carbons. Of the three carbons, there are different fractions in which this carbon could be labeled, either the first carbon or the second or the third. So we have like eight different variations in which A could be labeled. Now, if we have all those eight different variations, how will that get distributed to produce the different variations of C? C is one carbon, so either it could be labeled or it could not be labeled. That's the only case. Okay, so now we have to draw or try to relate how this conversion from A to C happens. Let me start drawing so it's easier. So first, yeah. So what we will have here is the different isotopomers of A. The first one we will have, so let this be the first way. Let this be the second one where this is alone colored. Here, this is alone labeled. Then here, this is alone labeled. Then in the next case, this is alone labeled. In the next case, this is alone labeled. In the next case, this is labeled. In the last case, So these are the different isotopomers for the substrate, where this is C1, this is C2, and the third is C3. Now this is substrate, whereas in the product you will have only two different variations. Now we have to see how each of these can give rise to either this or this. This is C1 alone. So now this is unlabeled. This is also unlabeled. Now unlabeled gives rise to unlabeled. That could be possible. So 1. But unlabeled cannot give labeled. That is 0. Labeled cannot give unlabeled. Labeled can only give labeled. Now, okay, one thing that one has to take into consideration here is there could be a possible error here. They have not mentioned which carbon from this is being given here. So it is given 1 and 2 from this is here, and this is also given as 1. So depending on which of these carbons is getting incorporated here, the values in the matrix would vary. But I will talk about the general procedure that is being used to form the isotopomer mapping matrix. So what we are assuming here is this C1, because it's mentioned as 1 here, this C1 is the one that is getting converted to C. Is that okay? So going by that, this empty would give this. So 1 again, and this unlabeled cannot give the labeled one. So 0 here. Uh, then this is unlabeled. Unlabeled giving an unlabeled, which is possible. But this unlabeled cannot give rise to labeled carbon. So that is not possible. Then this is a labeled carbon. Labeled carbon cannot give unlabeled carbon. So that this is 0.
So here we have to look for first carbon, whether it is labeled or unlabeled.
So the problem here with the question, the way it is given is They have not mentioned which carbon is getting incorporated. They say that there are three
So,
carbons, one, two, three, and they say that
uh,
in C it is one, but for B also they have given one and two. So I am not sure if we have to consider the third carbon or not. So I'm just talking about the general procedure in which the isotopomer
so
mapping matrix is derived. Yeah.
suppose if it is third carbon, it is mentioned third here
No, no. They
at
so
C.
if it is third here, they should have mentioned
Uh-huh.
third here. Like I think you can see my pointer here. Are you able to see
Uh-huh.
my pointer? So if they've
Yeah,
given
yeah.
it as three here, then we can
Uh-huh.
take the C3 here. You see this empty, 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 then
A.
filled, then empty, then three continuously filled. If they have mentioned
So
as three here, we can consider the third carbon here and we can derive it. We can still derive
so
it. It's not an issue. But they have not
so it
mentioned.
will be
Yeah,
so
sorry.
it will be 1 1 1 0 1 0 0 0 like that.
So we have two rows here,
Hmm.
right? So we have to fill for both the rows.
If I'm talking about the first row,
Okay, you're talking about the first row. 1 minute. Let me take
then
1 minute. 1 minute. Let me let me write this so that it's easier than drawing. Give me a minute. Okay, so we are talking about third carbon, is it? You want to
third
consider
carbon.
third carbon. Okay.
Yeah,
And
yeah.
here this is the
Hmm.
order in which it is there.
Yeah,
So
yeah, yeah,
third
yeah.
carbon. So yeah, it will be one and then one
One,
because three are empty. Then
then
the
zero,
fourth one will be zero.
then one,
Then again you have an 1, 2, 3, 4 is done. Then you have an empty, so that would give one.
one, then three zero,
Then you will have three, sorry, zeros in the
three.
first row. Whereas in the second row, you will have zero, 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 and then
One,
one, and then zero, and then one, and then one, and then one. Right. So if we take the third carbon, this is how it will be.
got it, got it.
Right. But they have not mentioned which carbon it is. So that is why there is some confusion in this particular problem. Was this clear, Ignatius? We'll again look at the answer that has been provided and then try to rationalize why it is this case. But if there is any confusion before that, you can tell me, Ignatius.
Yes. So, but what happens if I like interchange the carbon systems, like the labeling systems?
Sorry, your voice is breaking. Can you repeat?
Yes, sir. How would it affect the matrix if I interchange the labeled carbon positions?
So when you ask me how does it affect, can you like elaborate on that? I'm still not able to get your question.
Like suppose you in the matrix that you do, like we have C1, C2, and C3, right, in the column.
Right, we have C1, C2,
Right.
C3.
Now
Yes,
suppose in the first, second, third, fourth, suppose in the fifth place,
right.
I choose to label the first two instead of the, like I choose to label the second and third carbon instead of the first carbon.
Okay, okay,
Yeah, and like, I change the entire order after that.
right.
So like how will it affect the matrix there?
Okay, so you can understand it this way. All these matrices are for representation purpose. So I'll first tell you where this isotopomer mapping matrix is needed. We need this isotopomer mapping matrix when we have to get the isotopomer distribution vector. So this isotopomer mapping matrix is used as a function as one of the variables to find the isotopomer distribution vector. As long as the order here is conserved and the order in isotopomer distribution vector is conserved, when you multiply that, you would get the right fluxes. The idea is all the orders should be maintained in all the different matrices. Within the cell, none of this would affect. but mathematically it would affect since you will be considering a particular set of isotopomers for a particular set of intensities. That is the only place where this could go wrong. Otherwise, it wouldn't affect in any case. Did that answer your
Right, sir.
question or
And yeah, and the rows, like as you have depicted first the unlabeled and then the labeled.
yeah, yeah.
Yeah, so what happens if I switch that around? I first and then unlabeled.
No,
It makes
you're
my
one
row.
minute, 1 minute. It's slightly breaking. Can you repeat it? I got that first I represented unlabeled, then the labeled. I couldn't hear beyond that.
So what happens if I depict the labeled one first and then the unlabeled?
Yeah, yeah. So
So
that's
my
what I
rows
was
will
saying.
get interchanged.
So let's say if I put this first and this one, if we swap it here, what we have to consider is in the isotope distribution vector, which we previously were, when did we dis yeah, in this isotopomer distribution vector also, we have to ensure that the same order is being replaced. First, we start with the one carbon label, the second one is unlabeled. As long as it is the same in all the different matrices and vectors, calculations would lead to the right results. These are just representations, and the orders can be varied. But if you're varying it in one matrix, you have to vary it in all the matrices because this matrix gives you one information which is needed for construction of isotopomer distribution vector. If you have different orders in different matrices, when you multiply, you will be equating wrong equations, which would give rise to incorrect fluxes. Was
Right,
that clear?
right, correct. Yes, sir.
Okay, okay. Surender, was that clear? The procedure was it clear? We'll just look what has answer has been provided. Then we'll try to
Yeah,
rationalize.
yeah.
Yeah. Okay.
It is clear.
Okay. Okay. So they say the detailed solution is uh, in the detailed solution they say the answer is A. So they
Because
say
na,
that
I think
huh,
in two column, na, in two rows at the same position. For
sorry,
example, if we are talking about the first row,
first row, yeah.
option B,
Option B, yeah.
second point zero,
Yeah.
and second row, second point also zero.
Yeah. That
So
is wrong.
both zero cannot be happen.
Yeah, yeah,
Both
that
zero
is
cannot
wrong.
be happen.
That is wrong.
That is
Yeah, that is wrong. But otherwise, the other options A, C, and D, no, even D is wrong because we have another zero and zero here, like you have
yeah.
mentioned. But otherwise,
C also.
option
C also.
A, yeah, in C also we have 0 and 0. So the only possible way is option A. Yeah, that is one way to get the right answer, but that just depends on elimination. But what we have to understand here is how do we construct this isotopomer mapping matrix? That is important for the course. That's why we had gone through the example as to how to construct this. So, yeah. So the right answer is A because of other reasons where we have 0 and 0 in both the rows, which cannot be possible. Huh. So, hmm. So yeah, I hope this question was clear. Yeah. Now we'll go to the next question. Consider a MFA experiment using a glucose molecule labeled at the first carbon or a mixture of uniformly labeled 13C glucose. How many isotopomers can be found in a molecule consist of four carbon atoms? Okay. Now, what you can do is, like previously, we were able to do the isotopomer mapping matrix. You can, if possible, try to generate the set of isotopomers that can be produced out of this fully labeled, uniformly labeled glucose or the first carbon, and then how in a four carbon atom, I mean, in a four carbon molecule, this will be distributed. You can take a couple of minutes to solve this question, and then we'll see what the right answer is.
So
Yeah,
we can apply the formula 2 raised to power n. So if we are having the 4 carbon,
ha,
so 2 raised to power 4 is 16.
yeah. That is the number of possible
Uh,
isotopomers that one can have for four carbon molecule. That is true. Yeah.
uh So I think first statement is confusing.
You actually got the answer. So what you need to just understand a bit more is that uh,
Uh-huh.
independent of whichever carbon source that you use, whether it is uniformly labeled or labeled only one end, the number of isotopomers that a particular molecule of n number of carbons can possibly have is 2 power n only. Correct.
Mhm. Yeah.
So independent of the labeled substrate, it will only have 16 different isotopomers. And when it is having 16 different isotopomers, this uniformly labeled glucose would give rise to all the 16 isotopomers.
Yeah.
So there itself, we know we have like reached the answer. So the first statement is just an addition. This the intention of this question is just to see if we are able to get the 2 power n uh, relationship between the number of isotopomers and the number of carbon atoms. Yeah,
So here it is written na molecule is labeled at first carbon
yeah, first carbon.
or
Huh.
or or mixture of uniformly labeled means every carbon is labeled. All 6
Every
carbon are
carbon is
labeled.
labeled. Yeah.
So what we should consider?
So
First carbon or every carbon?
you can consider both cases. If since we have time, you can even try doing this mapping as to see how each of it would affect a particular isotopomer's formation.
So if we consider the glucose is labeled at first carbon,
Right,
so 2 raised to power n is equal to 16. It is not possible for isotopomers for 4 carbon molecule because if one carbon is labeled at one end,
right.
so in 4 carbon label molecule, all 4 carbons cannot be labeled.
That is where you have the uniformly labeled glucose, right?
uniformly labeled.
So they are
So,
saying a mixture, right?
it is
So
or a mixture like that.
sorry,
It is written or a
or
mixture.
yeah, it is written or a mixture of uniformly labeled glucose. So now your question is, if we are having only one labeled carbon, how will it give rise
Yeah,
to all
16
four
isotopes.
all carbons labeled?
Uh,
How
yeah,
I,
yeah. All four, yeah, all three.
yeah, how I have understood this question and how they have answered here also is the number of different isotopomers that can possibly be generated. So, because they have given this additional information that you can also include a mixture of uniformly labeled glucose, which satisfies producing all the 16 isotopomers, independent of whether they have mentioned this first carbon or this, what we have to focus is how many. Uh, what we have to focus is the relationship between the number of carbon atoms and the number of isotopomers. But answering your question, we will not be able to generate all I mean, 100% labeled four carbon molecule with just glucose molecule labeled at the first carbon. That is right. Your understanding is right. But what is expected out of this question is just the relationship between the number of labeled isotopomers and the number of carbon atoms.
Okay, I got it.
Huh. But if you're not convinced, we can have a discussion further.
No, I'm a little bit confused with the statement of the question.
Okay, it does not
Otherwise, there is
mean
no issue.
right.
Uh,
It's not worded properly. There should not
yeah,
be
yeah,
this,
yeah.
or maybe instead they should have given that you have a mixture of uniformly labeled carbon glucose and first
Yeah,
carbon glucose. Then they should have asked how many isotopomers are there.
yeah.
Yeah,
The statement is not correct, I think.
it's not worded properly. Yeah.
Yeah.
Was that clear, Ignatius?
Yes, sure.
Yeah.
And just one more thing.
Yeah.
I just had a doubt regarding the mapping matrix.
Okay.
So can you just revisit that maybe before the end of the session or now probably?
This question is it?
Yes, yes.
Yeah. We can discuss that if you want right now. Is it fine, Surinder, if we discuss it now?
No issue, no issue.
Okay. Yeah. Yeah. Go ahead with your question.
We have to say that it goes from A to C,
Yeah,
where
it goes from A to C.
A to C, where A is a carbon system and C is a one carbon system. So what if C is a two carbon system? Then how would the matrix map?
Okay. If C is a two carbon system, okay. What we have to remember here is, I hope you're able to see my pointer. If C is a two carbon system, you can tell me, you can tell me what are all the possibilities that we would have here. Uh,
Right.
1 minute. This will be C1, C2, and we'll have two unlabeled. So let's say we have two unlabeled, one labeled. Then in the last case, Both are labeled. Now we have to identify C1 to C1 and C2 to C2. So we'll know what are all the possibilities that we could get. So instead of this being an 8 cross 2 matrix, it will be a 8 cross 4 matrix. So that will be the difference. Does this clarify, or we can discuss further if it's
No,
not clear?
could we have an example? So, like, how would it be compared? So,
Okay, we'll try
like,
deriving it if that's uh, so. So we can do that here itself. So now C1 would go to this, C2 would go to this. Now we can discuss it further like we did it with Surinder. You can try answering which of these is applicable. Okay. So now C1 is unlabeled. Here it is unlabeled. For time being, let's not consider what the numbers that are given here. I'll remove them. Yeah. Now you can say this is unlabeled. C1 is unlabeled. C1 is unlabeled here. That is one.
yeah, so that would give us,
That is one. C2 is unlabeled. Here also C2 is unlabeled. Correct? So that will be,
yeah, one again.
huh. But in the next case, it's not that way. It would be, okay, here C1 is labeled. Here also C1 is labeled. So that would be 1. Here C1 is labeled. See, C1 is unlabeled. Sorry.
So it will be,
Sorry. I'll repeat that. I'll repeat that. So C1, C2, C3, C1 and C2. In the first place, it will be 1 because this can give rise to this. Then here it would be 0 because C1 is labeled whereas C2 is unlabeled. Then third case,
Yeah.
it's always 0. So all of these will be 0 because all of these have more than one labeled carbon. Whereas what we have here is 0 labeled carbons. So only the first one would have and one. Now we'll go to the second row. Now we have two, we have fully unlabeled, whereas here it is one unlabeled. So it would be 0. In the second case, it is C1. C1 is labeled. Here also C1 is labeled. So that would be 1. In the third case, C1 is not labeled, but C2 is labeled, which does not give rise to this isotopomer. So it would be 0. In the fourth case, C1 and C2 are unlabeled, but here we have C1 as labeled. So similarly, none of these match. Whereas in the third case, we have C1 is unlabeled, C2 is labeled. So comparing it with this, it's 0, another 0. The third isotopomer is where you have C1 unlabeled and C2 is labeled. So that would be 1. The fourth isotopomer We have C1 and C2 as unlabeled, but here we have it as labeled. So similarly, others do not match, it's 0. And in the fourth case, both are labeled. C1 and C2 are labeled. So first four do not apply, whereas in the fifth one, C1 and C2 are labeled. So 1 and then 0 and then 0 and then 0. So this is how the isotopomer mapping matrix would look like when we consider a 3 carbon substrate and a 2 carbon product. Is this clear?
Yes, sir. Yes, sir.
Right. Oh, okay. So we were discussing this question, MFA. Huh. So the next question is, during the 13 carbon metabolic flux analysis, GCMS is used to option A, measure the flow of isotope labeled atoms, option B, quantify the relative abundance of isotope labeled atoms, option C, measure the radioactivity of the isotope, and option D, all of the above. So we can just take a couple of minutes and then we can try with discussing this question.
So I think we should. It is all of the above.
We'll just 1 minute. We'll just take a couple of minutes and
Okay.
then we'll
Okay.
start with discussing why a particular option is right. Okay, we'll just give some time to think. Yeah, Surender. You can start with uh, explaining a particular option, and then we'll go with Ignatius. We'll take turns.
So, GCMS, it can be used how my labeled molecule is uh, flowing through the cell, in which pathway it is moving in and moving out. Okay. Also, we can measure the abundance also, how much labeled molecule is present in the cell, relative abundance. Also, radioactivity, because C13 is a radioactive material.
Hmm. Okay, so you're saying all of the
But
options given here are right. Okay, but sorry,
mm, uh, but radioactivity cannot be measured. I think.
we can't measure radioactivity. We need a Geiger counter to measure radioactivity,
We cannot measure the radioactivity.
but we can find differences in mass and abundance of differences, different isotopomers. That can be done. That is possible. And flow is usually derived out of abundances. We can't measure the flow also.
Okay.
In GCMS, we can only quantify abundances. Let's say I give 1 mole of glucose and I get X moles of a carbon product that is quantifiable through mass spec. Let us say, if that's the case, I can then say if this has been converted, X fraction of glucose has been converted to produce this carbon molecule, then this is the possible fluxes that that the cell will have in steady state to achieve this product formation. That's the only thing. So flux of all the metabolites within the entire pathway cannot be measured. But the only thing that we can measure is quantify the relative abundance of the isotope labeled atoms. Uh, yeah, so that is the thing. Uh, Ignatius, do you want to add on to this? Was the discussion clear or
I just had a doubt with option C, sir.
option C? Okay, yeah.
So measuring the radioactivity of the isotope implies the measuring the peak intensity, right?
Isotope implies measuring peak intensity.
The peak intensity. So isn't that possible through GCMS or NMR is used for that? Like how is the radioactivity actually gauged?
So radioactivity is not measured, but the fraction of compounds within the fraction of compounds that are radioactive within all the different isotopomers can be measured. We don't measure radioactivity,
Okay.
we measure masses. And since radioactive compounds are inherently differing in mass, that would give rise to different uh, products in mass spec, with which we get an idea about the distribution of the iso- isotope carbons. So we don't measure radioactivity directly through mass spec.
Okay, sir. Got it.
Yeah, yeah. So the answer is option B. The 13 carbon metabolic flux analysis is used to quantify the relative abundance of isotope labeled atoms. Yeah. The next question is, which of the following is not a biomass precursor? Option A, DNA, option B, RNA, option C, protein, option D, 2-phosphoglycerate. Yeah, Surendra or Ignatius, anyone would want to attempt answering here or trying to explain?
Because enzymes are responsible for making the products,
Huh. So,
and
huh.
protein products contribute to the biomass.
Right.
And enzymes are proteins.
Huh.
Proteins are encoded by the DNA with the help of the mRNA.
Right.
So all three they are responsible for biomass.
Huh.
Okay.
Right.
And 2-phosphoglycerate, it is not the means starting material for anything.
Hmm. Hmm.
So it is coming in between.
Hmm. Hmm.
So
So it
we
is
cannot
not an
say
exact
it.
precursor. Okay.
Yeah, it is not the exact precursor. So we cannot say it is contributing the biomass. Actually, glucose is contributing the biomass.
Right. Hmm. Hmm. Right. Yeah, that's the reason. Since proteins are enzymes that contribute to production of other stuff which are being encoded by DNA and RNA. The options protein, DNA, and RNA are uh, biomass precursors, whereas 2-phosphoglycerate, it is since it's only an intermediate, it cannot be considered as a precursor for biomass formation. Right. The next question is choose the incorrect statements for gas chromatography, mass spectrometry measurement, and MFA measurements. So the options are molecules are derivatized during GCMS, molecules get fragmented into different sized species, it cannot identify the position of labeled atoms. Option D, none of the above. Huh. So you can take a couple of minutes to answer this question.
Can I start?
Huh, you can try explaining the different options first.
Uh,
We'll discuss
yeah,
that
yeah.
and then we'll see which one is right. Yeah.
Uh, as gas chromatography,
Huh.
it is done for the volatile molecules.
Okay.
So if my molecule is non-volatile,
Ah,
or I can say it is a polar.
right. Polar molecules are usually non-volatile. Yeah.
So some their non-polarity is increased by adding non-polar molecule to them.
Basically, they are methyl groups. Right.
Uh, methyl group
Methyl
we
groups.
add them.
Right.
So it is called the derivatization.
That is derivatization. True.
Also, we cannot study the intact protein in the gas chromato or in the spectrometry, mass spectrometer. We have to break down it into the fragment. Then we have to analyze the fragments,
True. We
like their
want
sequences.
in case of proteins, what is being done is proteases like pepsin or trypsin is used to digest the protein so that you get different fragments of the same protein. Then this is being
Uh,
used in chromatography where you get mass for a particular peptide, short peptides instead of proteins. So they get fragmented into different sized species. True. Huh.
Overlapping fragments. No?
Right. Yeah, it is usually overlapping because the kind of proteases that is being used are usually non-specific. They can either just cut at different arginines or lysines instead of
Mhm.
instead of being able to cut at specific positions like stretch of amino acids. It just looks at one particular amino acid and cuts at it. And since in proteins you have repetition of amino acids, when it is being cut by a protease, you get overlapping fragments. Yes.
Also, mass spectroscopy cannot identify the position of the labeled atoms.
Hmm. Yeah. Is there a rationale to that?
Only deal with whether the molecule is labeled or not. That's all. So you cannot find the position of the labeled molecule.
Mm-hmm.
labeled atom, position of the
Mm-hmm.
labeled atom with the help
Mm-hmm.
of these techniques. So none of the above, in my opinion, all are
Okay.
the correct.
Okay.
Hmm.
Yeah. Would you want to answer why it can't identify the position of the labeled atoms?
Because these techniques they have to deal with the whether the molecule is labeled or not. That's all. Okay, if the molecule is labeled, then you can get the results. If the molecule is not labeled, we cannot get the results.
No, no, no. So,
No?
one thing that you'd missed is, uh, even if it is not labeled, you can still measure its mass. So,
Yeah,
one
yeah.
of the isotopomers that we always deal with is the unlabeled, right? It
Okay,
will be
okay.
unlabeled. So, we can still measure the mass, but in mass spec, independent of the position of the labeling, since only mass is being measured, all of these, even if they have a labeled isotope, like for example, this is C13, then C12 plus C12, then the difference in the mass between the three different isotopomers that are being shown here is zero. All of them have the same mass. So you will not be able to differentiate whether it came from this or this or this. That is why they say that it cannot identify the position of the labeled atoms. Although the answer that you mentioned that none of the above is correct, we have to understand the reason behind why it can't identify the position of the labeled atoms. Was it clear?
Yeah, yeah, yeah.
Okay. Yeah, Ignatius, do you have any questions? Was this clear? This
Yes, sir. Yes, it is clear.
okay, fine. Yeah, I'll just read through the answer. So what they say is first a carbon labeled substrate such as glucose, labeled with the first carbon, or a mixture of uniformly labeled and unlabeled glucose is used as carbon source. The patterns of isotope incorporation into intermediate metabolites, usually amino acids, are then analyzed using GCMS measurements. Molecules are derivatized during GCMS and may be fragmented into different sized species. For example, metabolite A here yields a 2 carbon and 3 carbon fragment. GCMS measures the fraction of abundance of fragments with different number of labeled atoms, but it cannot identify the position of the labeled atoms. Okay. It's a generic description. Okay. The next question is the solver used to optimize small network in MATLAB is option A ADMD, option B linprog, option C QtR, option D GNU Octave. Yeah, we can just take a couple of minutes and answer this question. Yeah, Surendra.
Actually, I don't have any idea about that.
Okay, so uh, Ignatius, do you want to share what you think about this or
Yes, I'll give it a try from the lectures that we have attended.
right, right,
So yeah, so in order to optimize this small network in MATLAB, I think it is linprog because I've seen the statement that linprog is used to optimize the F value.
right? Hmm.
Right, so I'm going to go with that linprog.
Okay, okay. Yeah. So, among the options here, LinProg is the right. It is just a linear programming solver. So, it is available on MATLAB. So, what we need to provide as input here is the stoichiometric matrix and which reaction we want to optimize. So, I'll just unshare and try opening the LinProg description here on the browser. Stay connected. Yeah, so what we see here is the definition of Lindbergh. So we know that in flux balance analysis, we are having equations as linear combination, sum of linear combinations for a particular reaction. When we have it such, in such a manner, we have a stoichiometric matrix which gives us the relationship between the metabolites and the reactions and how the metabolites are involved in a particular reaction. When when that is being provided along with the reaction that we want to optimize on, which is maximizing the flux through a
Sir, you're not
particular.
audible.
Sorry. Yeah. So in this particular page, we have the definition of linprog. I'll add it here from MATLAB. So that would give you different variations of linprog. It's basically a linear programming solver. What we have in our case of flux balance analysis is a network of reactions with metabolites participating in different reactions, where we represent the participation of a particular metabolite in a particular reaction in the form of stoichiometric matrix for all the reactions within the cell. Right. Then we also provide some inequalities as to how much a particular reaction can maximally have flux or minimally have flux, which are the lower bound and the upper bound. So what is generally used in our case is that A Lin-prog with F A B A equivalent B equivalent lower bound and upper bound. So A equivalent and B equivalent says gives you information on any equalities, which could be a particular value that this has to be of a particular value or not. And f is the function that we want, such that f prime cross x the it's being solved. So here A would be the matrix, B is 0. So if you remember the steady state condition that we follow, S dot V is equal to 0, it is similar here. So we'll have A as our stoichiometric matrix, B will be 0. We will find X here. So we will use this particular solver in MATLAB to solve for to identify different fluxes. Yeah, so that's how linprog works. Now let me go to the PowerPoint. So the answer is linprog is the solver that is being used. And the last question is, which of the following technique or techniques cannot be implemented to quantify the relative abundance of 13 carbons in MFA experiments? Option A, high performance liquid chromatography. Option B, gas chromatography or mass gas chromatography coupled with mass spec. Option C, nuclear magnetic resonance. Option D, all of the above. Yeah, you can take a couple of minutes and try to answer this question.
So HPLC cannot be used.
Hmm. Okay, whereas gas chromatography and NMR can be used.
Yeah.
Hmm. More or less, it's right because HPLC is based on both the charge and the mass, but not to the resolution of 1 Dalton, which is the difference between isotopes. So, yeah, HPLC cannot be used, right? So, in the answer, they say that in MFA experiment, a carbon labeled substrate such as glucose labeled at the first carbon or a mixture of phenol labeled carbon used as carbon. So, the patterns of isotope incorporation into intermediate metabolites are then analyzed using GCMS and NMR measurements, right? So, yeah, with this, we come to an end of the discussion for today's um, problem solving session. If there are any further doubts in the lectures, you can post it now. We can have a discussion since we have about 3 minutes left.
So last time we discussed the basis vector.
Yeah, basis vector. I could not
We
solve
could
that.
not find the you could
Yeah,
not find the correct one.
I could not do that. I would present the answers. We would still have one-to-one discussion sessions after the eighth week. Today is the sixth week. After seventh week and
Mhm.
say eighth week, we will still have a one-to-one discussion session. If I'm unable to do it before the eighth week, we'll have it solved during the discussion session. In the discussion session, I'll be summing up all the lectures giving an overview of the courses. So I'll try to solve the answers during that discussion session, if that's fine.
Okay.
Right. Yeah, Ignatius, if you
Okay.
have any questions.
Thank.
So is exponentially considered a quantitative or a qualitative technique?
No, no, flux balancing is considered quantitative when all the constraints are taken from experimental data. So the constraints that you give for your flux balance equation for your solver, if you take it from experiments, then the solver can predict the fluxes, which would more or less match what is being observed in the experiment. So it is considered as quantitative, but one has to also ensure that whatever constraints are given is matching experimental data.
Okay, sir.
Yeah. Okay. Yeah. If there are no more doubts, we can close the session for now. Okay. Thank you. Thank you, Ignatius.
Thank you, sir.